Hello everyone, my name is Apu and I'm a PhD student at the University of Groningen. And today I would like to talk to you about our research titled Movement Tendencies in Wild Migrant and Resident Sticklebacks, a mesocosm experiment done by me and Jacob Gisman at the University of Groningen, the Netherlands. First, I would like to explain to you our system. The river systems in the north of the Netherlands harbor several populations of sticklebacks that are traditionally migratory, and they migrated to the sea in autumn and return to the rivers in spring in order to breed. However, this pattern has largely been shut off in many cases due to water management effort through construction of structures such as pumping stations or weirs across these rivers. In this map, we see that the green area represents the river systems that are open to the sea, while the red regions represents parts of the river that are completely blocked off. And in addition to this, much of the network of side arms of the river are completely blocked off from the sea. So this has led to a unique situation of replicate natural experiments over 50 years ago due to water management effort, leading to resident populations across these blocked arms of the river. So our main question that we address in our lab is how has these human-induced changes affected morphology and behavior of sticklebacks? First, we address this in a lab-based system where we saw immediately that in terms of morphology, migrants and residents are very different, with residents being almost half the size of migrants. In addition to this, we conducted several behavior assays in the lab where we also saw that migrants and residents form two separate clusters across these important behavior traits. In conclusion, from these controlled lab experiments, we see that residents are consistently smaller, bolder, more exploratory, more active, more aggressive, and also show less than migrants. However, in this case, we did not explicitly test long distance movement tendencies, such as those you would expect for migratory behavior. So this leads to our current study question, which is whether the residents have lost their migratory tendencies or their larger distance movement tendencies. And how do we study this? It is not possible to do this in the field because following such tiny fish over long distances is not possible yet. Also in the lab, the scale is too small for concluding effectively about long distance movement tendencies. So we constructed at the University of Groningen a network of connected ponds that allows us to monitor fish over smaller scale, that is within a pond, and also over larger scale between the ponds in order to effectively study these movement behavior in these mesocosm experiments. Let me explain to you further how we study this. We use a system of pit tags and pit tag antenna, which means a passive integrated transponder is injected into the body cavity of the fish, and we recognize or we detect these fish when they pass through antennas. And in order to study smaller scale within pond movement, we have the setup of bottom antennas, which would give us a score about exploratory type behavior in our sticklebacks. And also we have a framework of surface antennas, which leads to movement tendencies related to risky behaviors or boldness in sticklebacks. And these two systems were present within one pond. And in order to quantify larger scale or bit movement tendencies, we look at movement between ponds over scales of 15 meters, where we detect fish when they cross between ponds by equipping the corridors with pit tag antennas. So to get a proxy for their movement tendencies, we quantified crosses between bottom antennas and between top antennas as a proxy for smaller scale within pond movements. And to quantify a larger scale movement, we quantified crosses between these ponds that a fish makes throughout the experimental duration. Now we get to the results. Here we represent a plot of the frequency distribution of the number of crosses that a fish makes. So here in our case, for exploratory type of crosses or the smaller scale movement tendencies, we see that migrants and residents are not very different, and similar sort of pattern arises even for the movement tendencies across the surface antennas. So this leads us to a conclusion that residents and migrants have similar movement tendencies for smaller scale within pond movement. However, when we looked at the movement tendencies between ponds, in terms of crosses between ponds, we see that migrants made much more crosses than residents did, and residents moved significantly less between ponds than migrants in terms of these larger scale movement behavior. In addition to this, we tried to quantify how many of these fish, how long it took fish to exit the first pond in order to move across these ponds and show these large scale movement tendencies. And here we see that there is a significant proportion of non-movers or fish that did not exit the first pond in residents as compared to migrants. In addition to this, we quantified whether the larger scale movement tendencies between ponds is repeatable or not, which means whether individuals that move less consistently move less across different days. And we see clearly that across 2 days, we did this over 4 days, and I'm representing 2 days for simplicity. We see that individuals that moved less consistently moved less, and individuals that moved more always moved more, which is given by the Spearman correlation of 0.7. So we see that between pond movement is highly repeatable. This leads us to our conclusion that migrants and residents differ consistently in larger scale movement, but not in small scale movement. And 50 generations of human-induced isolation is sufficient to cause substantial differentiation in morphology and behavior. And in addition to this, through our experiments, we see that mesocosms are a promising method to bridge the gap between field and lab-based studies. With this, I would like to thank Jacob Hisman, who conducted the study along with me, our supervisors, and all of our collaborators, and you for your attention. Thank you very much.